Hello students, the topic of my today's lecture is optimization techniques. Now let us understand the definition of optimization. The word optimize simply means to make as perfect, effective or functional as possible. However, today the term implies that computer and statistics have been utilized to achieve the objectives. Now with respect to the drug formulation or pharmaceutical processes, optimization is a phenomenon of finding the best possible composition or operating condition. Accordingly, optimization has been defined as the implementation of the systematic approaches to achieve the best possible combination of product and the process characteristics. Now, what is the necessity or why the optimization is necessary? The primary objective may not be to optimize absolutely, but to compromise effectively and thereby produce the best formulation under a given set of conditions. So these are the four major advantages. We can reduce the cost, we can save time, we can produce the product in a reproducible manner, we can induce safety, and we can reduce error in the production processes. Now, what are the optimization parameters? Parameters may be the problem type, may be constrained, unconstrained, dependent, or independent. And problems which are under our control are called as constrained. The problems which are not in our control are called as unconstrained. And variables are dependent and the independent variables. Now let us understand what are the variables. Design and development of a drug formulation or a process usually involves several variables. The input variables which are directly under the control of the product development scientist are called as independent variables. जो हम साइंटिस्ट हैं फॉर्मुलेशन बना रहे हैं जो हमारे कंट्रोल में वो कहलाते हैं आपके इंडिपेंडेंट वेरिएबल्स. यानी एक्सिपिएंट का अमाउंट हमारे कंट्रोल में है, कितने फोर्स से हमें टेबलेट को कंप्रेस करना है वो हमारे कंट्रोल में है, कितना टाइम हमें अपने फॉर्मुलेशन को मिक्स करना है, वो सारे कहलाते हैं इंडिपेंडेंट वेरिएबल्स. These variables can be quantitative depending on the quantity, like percentage of the excipients, कितना amount of disintegrant add कर रहे हैं, कितना temperature हम रख रहे हैं, time कितना लग रहा है जिसे हम measure कर सकें, वो कहलाते हैं आपके quantitative. दूसरे होते हैं qualitative on the other hand, which include the type of emulsifying agent, the type of solubilizer, the type of the tablet punching machine. Independent variables which influence the formulation characteristics और आउटपुट आर कॉल्ड एज फैक्टर्स. यानी सारे वेरिएबल जो इंडिपेंडेंट है वो फैक्टर्स नहीं है, जो कि हमारे फॉर्मुलेशन की कैरेक्टरिस्टिक्स को इन्फ्लुएंस करेंगे, वही कहलाएंगे आपके फैक्टर्स. फॉर एग्जांपल द टेम्परेचर, लाइक 30 डिग्री टेम्परेचर, 50 डिग्री, दिस द लेवल्स फॉर द फैक्टर दैट इज टेम्परेचर. नाउ the characteristics of the finished drug product and the in-process material are known as the dependent variables. जो हमें प्रोडक्ट बनाया, जो उसकी कैरेक्टरिस्टिक्स हैं, जैसे ड्रग रिलीज क्या आ रही है, उसकी फ्राइबिलिटी क्या आ रही है, टैबलेट ग्रेन्यूल्स का साइज कैसा आया, टैबलेट का डिस्टिलेशन टाइम क्या है. Now these are called as dependent variables. Popularly they are also called as the response variables. These are the measured properties of the system to estimate the outcome of the experiment. Usually these are the direct functions of any change in the independent variable. Now the effect and interaction. The magnitude of the change in the response caused by varying the factor is termed as an effect. यानी हमने अगर एक्सिपेंट की कंसंट्रेशन चेंज की, उससे हमारे ड्रग के फ्राइबिलिटी में या ड्रग के डिस्टिलेशन टैबलेट के डिस्टिलेशन टाइम में क्या चेंज आया, that is called as effect. The main effect is the effect of a factor arranged over all the levels of other factor. Suppose we are taking two factors. One is the excipient concentration. Its own effect is called as the main effect. However, an interaction is said to occur when there is a lack of additivity of factors effect. Suppose there are two factors, amount of excipient and the mixing time. And both these factors, if they are affecting each other, then there will be that is called as interaction. Now the levels of the independent variables of the factors can be coded as -1, 0 or +1. The lower is -1, the middle or the intermediate range is 0 and the highest range is called as +1. For example, if you are taking the starch as a disintegrating agent and we are taking 3 levels, 5 to 10. So 5 is the lowest, it is called as -1. 7.5 will be in the middle, it will call as 0, and 10% is the highest, it will call as the +1. So -1, 0 and +1. It is called as the coding of the levels of the independent variables. Now the run of the trials, the number of experiments we have to conduct as per the experimental design are called as run or trials. And next is the principle of randomization, that is the manner of allocation of treatment to the experiment units, and replication, the number of units employed for each treatment, and error control or the local control, that is the group of specific types of experiment to increase the precision. Now the data which we get after the experiment has been conducted can be visualized in terms of 3D models. They are called as 3D graphical representation of the response plotted between the two independent variables and one response. Suppose we are taking two independent variables like the concentration of a polymer 1 and polymer 2 and the zeta potential, which you can see in the picture. So the two excipients are the independent variables and the zeta potential is the dependent or the outcome or the response. So in this way we are showing a relationship between the two independent variable and the one dependent variable in terms of a 3D contour plot. This is called the 3D response surface plotting. The same thing can be represented as a two-dimensional, that is called as contour plot. So contour plots are the two-dimensional, and in the previous slide which I told you is the three-dimensional view, that is called as 3D response surface plots. Now we can also define or we can explain the effect of the response and the independent variables as a mathematical equation that is called as the polynomial equation. These are some of the equations which you can obtain after we are showing the relationship between the dependent variable and the independent variables. Now, how to study the factors? First, we have to conduct the systematic screening, and we have to also understand the influence of the factor studies are carried out before the design of experiment optimization. Screening means we are separating rice from the rice husk. See, the main thing is that all the independent variables cannot be called as a factor. Only those independent variables can be called as a factor if they are affecting the dependent variable. For example, हमने टेम्परेचर का इफेक्ट ले रहे हैं, हम देख रहे हैं कि टाइम बढ़ाने से क्या हो रहा है, स्पीड बढ़ाने से क्या हो रहा है. अगर स्पीड बढ़ाने से हमारी फॉर्मुलेशन की कैरेक्टरिस्टिक्स पे कोई असर नहीं पड़ रहा, तो हम कह सकते हैं कि जो हमारी स्पीड ऑफ रोटेशन है वो फैक्टर नहीं है. जबकि टेम्परेचर और टाइम जो हम ले रहे हैं उसका असर अगर पड़ रहा है हमारे रिस्पॉन्स पे, हमारे फॉर्मुलेशन की कैरेक्टरिस्टिक्स पे, तो हम कह सकते हैं ये दो हमारे फैक्टर्स हैं. तो इस तरह से हमें बहुत सारे वेरिएबल्स में से उन्हीं को चुनना होता है जो कि हमारी ड्रग की फाइनल कैरेक्टरिस्टिक्स पे असर डाल रहे हैं, उन्हीं को हम फैक्टर्स कहते हैं. इसके बाद हम देख सकते हैं कि और स्टडी कर सकते हैं कि फैक्टर इन्फ्लुएंस, इसमें हम क्वांटिफाई करते हैं इफेक्ट ऑफ फैक्टर्स एंड उनके बीच में क्या इंटरेक्शन हो सकता है, यानी किस लेवल में हमें फैक्टर्स लेना है, किस परसेंटेज में हमें एक्सिपेंट्स को लेना है, क्या हम मिक्सिंग टाइम रखना है और इन दोनों का आपस में क्या इंटरेक्शन आता है. ये कुछ स्टेप्स हैं. हम एक पिक्चर से वी कैन अंडरस्टैंड द फर्स्ट स्टेप. यू कैन सी फ्रॉम द बिलो. We have to define the objectives of the study and plan the experiment. Then after that, step 2, we have to screen the factors and factor influence studies. Second is we have to perform the response surface methodology using the experimental design, means on a small scale, on the lab scale. Step 4 is formulation and evaluation of the drug delivery systems as per the experimental design. We can also use the computer aided modeling and search for an optimum requirement of the factors. Then we have to validate the design of experiment, and finally in the step 7 we can scale up and implement the pharma production cycle. So I suppose you must have understood the basics of the optimization techniques. Thank you and have a nice day.